NBSP, an amphoteric molecule can either donate or accept a proton, so can act either acid or base. At low pH more proton environment carboxyl group accept proton and became uncharged and overall charge is positive. Whereas at high pH less proton environment amino group loses proton and became uncharged and overall charge became negative. At specific value of pH called isoelectric point, pi, every amino acid exists as dipolar ion or Zwitter ion. A Zwitter ion is a compound with no overall electric charge but contains positively and negatively charged groups. What is optical activity of amino acids? Amino acids except glycine are optically active. They rotate the plane of polarized light. Optically active molecules contain chiral carbon. A tetrahedral carbon atom with 4 different constituents are called chiral carbon. Optically active compound having chiral carbon rotate plane of polarized light to clockwise are said to be dextro-rotatory and designated as plus or D. Whereas optically active compound having chiral carbon rotate plane of polarized light to anti-clockwise are said to be levorotatory and designated as or L. Optical activity measured by polarimeter, and it's a quantitative measurement. Magnitude of optical rotation indicates extent of rotation and sign represent direction. Optical activity or rotation depend on concentration of compound, temperature, wavelength of light used, solvent used to dissolve, and light path. Optical rotation of solution at a given temperature and wavelength is A degrees equals alpha T lambda Cl. A degrees equals observed rotation. C equals concentration of solution in G/ml. L equals light path length in decimeter, dm. NBSP, alpha T lambda equals specific rotation at wavelength, nm, and temperature, Celsius. Let's talk about specific rotation. Specific rotation is a